Hi, I'm Jessica Walden, and I contributed to a 24-hour field study to elucidate the Diel cycle of monomethylmercury in a tidally influenced coastal lagoon system in Southern California. Mercury is a potent neurotoxin that comes from both natural and anthropogenic sources. Human activities have almost tripled environmental mercury levels over the past century, and different mercury species exist in the environment, and transformation between the species is mediated both biotically and abiotically. Methylmercury is the form that can be taken up by primary producers and bioaccumulates to higher trophic levels. The largest bioaccumulation step in methylmercury uptake occurs via primary producers. Concentrations increase by 100-fold from ambient water levels to levels within cells. Different bodies of water exhibit unique biogeochemical and hydrological characteristics, which in turn contribute to varied mercury cycles. Sulfur and iron-reducing bacteria methylate different mercury species, but their presence and metabolisms are affected by chemical and hydrological characteristics of the ecosystem. Methylmercury can be demethylated via photodegradation, which is an abiotic process, or by organisms. The concentration and type of dissolved organic matter and dissolved organic carbon can also influence ambient water concentrations of methylmercury. They are sorbents of both methyl and regular mercury and also attenuate light. Previous studies have focused on either saltwater or freshwater systems, but not on systems that fluctuate between the two. Because San Elijo is a tidally influenced system, it will exhibit characteristics of both freshwater and saltwater environments depending on the tide. We hypothesized that methylmercury levels would be driven by factors that control oxygen availability, like water quality, photosynthesis, and respiration, and that methylmercury production would vary over the Diel cycle depending on the photoperiod and the tidal cycle. San Elijo is the terminus of Escondido Creek and drains into the Pacific Ocean at Cardiff Beach. We sampled every 1 to 2 hours over a period of 20 hours. We used a sonde and implemented water quality probes at surface and depth to measure biogeochemical properties. We collected water samples to obtain methyl and total mercury concentrations, nutrient concentrations, and phytoplankton cell counts. Turbidity peaked at low tide and pH peaked at high tide, and dissolved oxygen concentration was highest at daytime high tide. Nitrate concentrations were below detection limits, while both phosphate and ammonium peaked at low tide and troughed at high tide. Picophytoplankton abundances were highest during the day when we began sampling, and plummeted at night and remained low throughout the rest of the study. The phytoplankton community structure and photosynthetic efficiency were influenced by irradiance over the Diel cycle, which is expected. Tidal flow brought seawater with high concentrations of dissolved oxygen into the lagoon, but low tide brought fresh water with low concentrations of dissolved oxygen. In addition to the tides, biology has an effect on the dissolved oxygen concentrations through a balance between photosynthesis, which adds dissolved oxygen, and respiration, which removes it. Daytime photosynthesis dominated by contributing relatively high concentrations of dissolved oxygen in spite of a low tide. However, at night, the influence of respiration depends on the tidal cycle. Seawater has higher dissolved oxygen concentrations, so nighttime high tides will have higher concentrations of dissolved oxygen than nighttime low tides. 4 primary sets of conditions emerged from observing the overlapping tidal and Diel cycles, as seen in Figure 1. At high tide during the day, dissolved oxygen is high and chlorophyll is low because seawater brings with it higher levels of dissolved oxygen and photosynthesis adds to the pool. As night falls, dissolved oxygen drops. This is because rates of photosynthesis are declining. As the tide goes out and the lagoon becomes more freshwater influenced, chlorophyll levels slightly rise and dissolved oxygen reaches a minimum due to a lack of seawater influence and photosynthesis throughout the night. As the sun rises, chlorophyll levels rapidly increase, photosynthesis drives up dissolved oxygen concentrations, and this overcomes the influence of the low tide. Because there are 4 primary conditions that are heavily dependent on tidal and light cycles in the lagoon, we expect our mercury results to follow a similar cycle. We are waiting for the results of our mercury analyses. The methylating bacteria thrive in anoxic conditions, so we might see high methylmercury concentrations after the nighttime low tide, and photodegradation and phytoplankton uptake should drive mercury concentrations in the ambient seawater down during the day. We hope this research will both contribute to the pool of studies on mercury cycles in other aquatic environments and also help elucidate the factors that contribute to mercury bioaccumulation. Thank you.
That was phenomenal.
Okay.